Hello students, we had seen the pre-fertilization events of angiosperms in the previous lecture. We had seen the structure of the flower, structure of the microsporophyll, megasporophyll, and how the microsporophyll produces the microspores and how the megasporophylls produce the megaspore. We had also seen the development of the megaspore into a matured female gametophyte. In case of the angiosperms, I told you that the female gametophyte is 7-celled, 8-nucleated, and it is called as an embryo sac. In this lecture, we will see the fertilization and the post-fertilization events that happen in angiosperms. This is the ovule, and inside it, the female gametophyte has matured. Okay, so the embryo sac is ready with the female gamete. Now there is pollination. Pollination occurs and the pollen grain lands onto the stigma of the flower. There is a brief communication between the stigma and the pollen grain, and once the pistil accepts this pollen grain, one of the two synergid cells of the embryo sac will break open and will release a chemical which will create a chemical gradient all along this path. The contents of the degenerated synergid cell will create a chemical gradient across all this path. The pollen grain germinates and will produce a pollen tube. So let's say this is the pollen grain and it has germinated. This is the pollen tube. Right at the tip there is one nucleus which is called the tube nucleus, and behind that there will be two male gametes. These are the male gametes and they will be non-motile. Bryophytes, pteridophytes, they all had motile gametes. Some of the primitive gymnosperms also had motile male gametes which started becoming non-motile in the higher gymnosperms. This pollen tube is going to move toward the increasing concentration of the chemical released by the synergid cell. There will be a chemotactic movement there. There will be chemotaxis. It will come down into the ovary and take a turn and enter inside the microbiome. So over here in this diagram you see that it has entered the microbiome. The moment the pollen tube enters inside the microbiome, the two polar nuclei here, remember the polar nuclei? The two nuclei in the center are known as the polar nuclei. The moment the pollen tube enters inside the microbiome, the two polar nuclei of the central cell will fuse together to produce a 2n nucleus. This 2n nucleus is known as the secondary nucleus. So right now the situation will be that the pollen tube has entered inside the microbiome and the two polar nuclei no longer exist, but instead exist a 2n nucleus. This will be the secondary nucleus. Let us say this synergid cell was the one which had degenerated. The pollen tube will extend toward it and it will enter it. There is certain mechanism inside these synergid cells which will lure the pollen tube towards itself, and it will help to break it open once it enters inside. These finger-like projections that are there inside the synergid cells are known as filiform apparatus. Okay, filiform apparatus are present in synergids, and what do they help in? They help to release male gametes. So the filiform apparatus will crush the tube open, the two male gametes will be liberated inside the embryo sac. The first male gamete is going to come and fuse with the egg cell. The second male gamete will fuse with the secondary nucleus. There will be not one but two fertilizations in the angiosperms. Very, very important. This is called as double fertilization, which is a characteristic of only the angiosperms. Okay, there will be not one but two fertilizations here. Alright, in gymnosperms there was just one fertilization of the male gamete with the female gamete. Alright, over here there are two fertilizations here. This phenomenon is known as double fertilization. Over here, two fertilizations have taken place. Let's mark them as 1 and 2 and let's discuss. The first fertilization over here has taken place between the first male gamete, okay, which is N, along with the female gamete, which is also N, and the resultant is a 2N zygote. This first fertilization is known as syngamy / fertilization. There is a second fertilization that has also taken place. This has happened between the second male gamete plus secondary nucleus here. The second male gamete is N and the secondary nucleus is 2N, and the resultant of this is a nucleus which is called as the primary endosperm nucleus, short formed as PEN, whose ploidy is going to be 3N because this is N and this is 2N. This fertilization is known as triple fusion. Why triple? What is so triple about it? Because 3 nuclei have fused together. That's why triple fusion. What are the outcomes of these 2 fertilizations? The first fertilization that has formed the zygote. This zygote is going to eventually grow into the new sporophyte. New sporophyte. Ploidy will be 2n. This will form the embryo of the new sporophyte. So that is your new sporophyte whose ploidy is going to be 2n. The 2n plant has formed a new 2n plant again. Fine. What about the second fertilization? The second fertilization, which produces the primary endosperm nucleus, is the one that will divide mitotically and will produce a triploid endosperm. Endosperm, a nutritive tissue as seen into a corn seed. Okay, very clearly. This is the embryo. This is the endosperm. Endosperm will provide nourishment, nutrients to the growing embryo when it is in the seed stage. This will produce endosperm whose ploidy is going to be triploid. Very important. The endosperm of the gymnosperms was formed before fertilization. This one is produced after fertilization. The endosperm of the gymnosperms is haploid. However, here the endosperm is triploid, not haploid. What will happen once these 2 fertilizations have occurred inside here, inside this ovule? Post fertilization, this ovule turns into seed. What about this ovary? This ovary will produce a fruit around it. Ovary produces fruit wall. Fruit wall is also called as the pericarp, and as such this will be your seed. Typical seed, there will be 2 layers of the seed coat. Okay, outer testa, inner tegmen, both will be together called as the seed coat. Okay, there will be an embryo here, and surrounding that will be an endosperm which will be 3L. In general, this is how the seed is going to be post fertilization.